So hello students and welcome back to another video tutorial by Genome Biotech. In this video tutorial, we would be looking at the end replication problem. Now, what is this end replication problem? The end replication problem refers to as the problem associated with the DNA polymerase in completing the process of DNA replication by replicating the telomeric genomic DNA. You know that telomeres refers to the end of the chromosome molecules. Okay, so now when you talk about telomere, when you are going to talk about, you're going to talk about the ends of the chromosome. If this is a chromosome, then the short arm of the chromosome, there are two arms of the chromosome, the short arm of the chromosome being referred to as the P, the long arm of the chromosome being referred to as the Q. The end point of the chromosome which is present is referred to as the telomere. This is telomere, which will be replicating the ends of the chromosome. Okay, now how does this telomere act, in what manner telomere function, and how does this telomere catalyzes the replication to effectively take place in order for the ends of the chromosome to be replicated? How does telomerase act? Now telomerase, as you all know, is a very special enzyme. Okay, the special enzyme called as telomerase is employed to catalyze the replication at the telomeric end. So, a very special enzyme is required to replicate the DNA at its ends, and that replicating enzyme is referred to as the telomerase enzyme. Okay, telomerase enzyme is actually a ribonucleoprotein. This is a ribonucleoproteins. That means it is an association of rRNA. It is an association of RNA and protein together with each other. When RNA gets associated with protein, then it leads to the formation of ribonucleoprotein. RNA plus protein will lead to the formation of the ribonucleoprotein that is being produced. This is ribonucleoprotein that is going to be produced by the combination of RNA as well as protein molecule. It contains RNA as well as protein. It is actually an example of reverse transcriptase because it has the ability to synthesize the DNA molecule using RNA as a template. So it synthesizes DNA molecule using RNA as a template. Okay, so this is telomerase is a very special enzyme because it is a reverse transcriptase. It is a RNA dependent DNA polymerase. RNA dependent DNA polymerase. DNA polymerase, and this RNA dependent DNA polymerase works in a template independent manner. It do not require any template. It works in a template independent manner. What do you mean by template independent manner? Template independent manner means there is no requirement of any template in this particular reaction. There is no requirement of any template. No template is being required in the entire process. Why? Because what happens here is that now there is no template which is being required. RNA polymerase, the reverse transcriptase, the telomerase itself has a particular template. If you look at the structure of the telomerase now, you will find that the telomerase has a tank shaped structure. Its structure will be something like this. This would be the structure of the telomerase enzyme. And telomerase enzyme here will have its own sequence. Okay, it will have its own sequence present here. And this own sequence of the telomerase enzyme will ensure that the effective base pairing is going to take place, and new nucleotides will be available for forming the base pairs. How? This is the telomerase that would be acting. This is the telomerase which has an RNA sequence TTAGGG. Okay, it has an RNA sequence present within itself, and that RNA sequence, see, this is an RNA sequence. This RNA sequence will provide the complementary. This is an RNA sequence which is present. It contains a sequence, that is RNA sequence. As it contains a RNA sequence, and this RNA sequence will provide the template for the biosynthesis of new nucleotides. See, this is what happens with our t-shirts, with our bushirts, etc. What happens with the t-shirts? What happens with the collar of your t-shirts or your shirts? the collar is being made to fold so that the ends are not being distorted, not being disturbed. Okay, so what happened with the case of chromosome? You know that if this is the structure of the chromosome, I'm just making the structure of a chromosome. Okay, if this is the structure of the chromosome, then if you leave the end of the chromosome as such, then these ends of the chromosome will be subjected to degradation by the exonucleases. These will be subjected to degradation to degradation by exonucleases. Exonucleases are the enzyme that will be cleaving the DNA from its ends. Okay, subjected to degradation by exonucleases. Here also, exonucleases based degradation, nuclease based degradation. Now, supposingly, if degradation is going to take place, then the DNA will be lost. DNA will lose its integrity. In order to protect this DNA molecule, what is being done? There are nucleotides which are going to be added over the ends of the DNA, and then the DNA is made to be folded. How the DNA is made to be folded? Please look here. There are nucleotides which are being added. So I'm just taking another color in order to possibly add few number of nucleotides. See this DNA, a number of nucleotides will be added, and then what will happen? It will be made to fold just like this. This is what we used to do with our collars. Our collars are being folded in order to protect our collars, from degradation. And here we will bind it with another proteins, and the proteins that are going to bind here is going to be and going to seal this process is the telomerase binding protein. We are going to seal it with the help of certain protein molecules. The protein molecules with the help of which we are going to provide a sealing is referred to as the telomerase binding protein. These are the protein molecules which are present. The protein is called as telomerase binding protein. These are the proteins which are present, and these are telomerase binding protein. Okay, these are telomerase binding protein. These are telomerase binding protein that are going to bind with the telomerase. Okay, this is how telomerase is going to bind. And when the telomerase is going to bind, it will hold the double stranded DNA molecule in this perfect orientation. This is how double stranded DNA molecule will be held. Okay, the mechanism just describes ensure that the 3' DNA end at each telomere is always slightly longer than the 5' end, which has been which with which it is being paired. See, the 3' end is being extended and then made to fold. It will form two different loops. One is referred to as the D loop, another one is referred to as the L loop. There are two loops which are being produced. One is called as D loop, another loop which is being produced is referred to as L loop. So these are the two loops which are being produced. A D loop system and an L loop system is being produced. Okay. Next, the mechanism described here ensures that the 3' end of at each telomere is always slightly longer than the 5' end with which it is paired, leaving a protruding single-stranded end of the DNA molecule. Now, this is the telomerase enzyme. This is an RNA-dependent reverse transcriptase enzyme. It will use the RNA sequence present over itself, will synthesize new DNA molecules. So, what are these? These are DNA. This is single stranded DNA. And what was the template which was being used was an RNA. So the enzyme reverse the enzyme telomerase is actually an example of reverse transcriptase. It is an example of reverse transcriptase. Okay, reverse transcriptase. Reverse transcriptase. This is an enzyme which is present. It is the enzyme that is present. Okay. So reverse transcriptase is going to be functional, and it is going to add nucleotides. Then the DNA is going to be polymerized. This is how the DNA polymerization will take place and the folding of the DNA will occur. Now, how the entire process takes place, which are the proteins, telomerase binding protein, how does these telomerase binding protein binds, that we must visualize now through an animation. Okay, so look at the animation which is being shown to you. Okay, please pay your attention here.
DNA polymerase
Just a second. This is the action of telomerase. We would be talking about what is the end replication problem, how the action of telomerase actually takes place, in what manner the telomerase act. Okay, are you able to visualize the screen in front of you? Okay, this would be the action of telomerase. We would be looking at how the telomerase of an enzyme solves the end replication problem. So what is this end replication problem? End replication problem refers to a problem that is being encountered when the cell is going to be replicable. When the cell is undergoing replication, you can just look here. Now, supposingly the cell has its genetic material in the form of the DNA molecule. This DNA molecule is undergoing replication. Okay, this is a DNA molecule that is undergoing replication. What happens in each replication cycle? Because the cell DNA ends are free, so they may be subjected to degradative action. The cell, the dsDNA, the dsDNA ants might be subjected to degradation via the endonuclease, via the exonuclease of the cell, via the exonuclease of the cell, via the exonucleases of the cell. Okay, that will be degrading the dsDNA from its ends, thus shortening the DNA molecule. So if such type of things will continue, then the DNA will be losing its integrity and will be lost. Thus, if it continues, the integrity of the dsDNA will be lost. Okay. So to maintain it, the action of the telomerase is of significant importance. Now, what is the importance of the telomerase enzyme? Telomerase will help in maintenance of the end of the chromosomes. So what does telomerase actually do? Telomerase maintains the ends of the chromosome. Telomerase maintains the ends of the chromosome, maintains the ends of the chromosome. Okay, this is what does the telomerase is going to do, and this would be of the most important significance in maintaining the ends of the chromosome of the cell. Now, how this process is going to take place, that we must understand. Okay, please look here.
requires an RNA primer to initiate DNA synthesis. This requirement creates a dilemma for the replication of the ends of linear DNA molecules. When you have completed this exercise, you should understand the end replication problem and understand how eukaryotes use telomerase to solve the end replication problem.
Okay, so you must know what is this end replication problem, and we should know that how telomerase enzyme is being used in order to resolve this end replication problem. So please move forward.
During replication, the lagging strand is synthesized as short fragments called Okazaki fragments. The RNA primers are removed by RNase H, filled in by DNA polymerase, and ligated by DNA ligase.
So this is what we have already seen. The primer is being provided by primase. The DNA is being biosynthesized in order to form the Okazaki fragments. The repair process is being done with the help of DNA polymerase 1. We have already seen about the Kornberg polymerase. How does Kornberg polymerase acts? And lastly, the ligation is being done with the help of another enzyme called as DNA ligase. So now we are going to move forward and understand the end replication problem.
However, DNA polymerase is only able to add DNA to the 3' end of a polynucleotide. Therefore, even if primase is able to synthesize an RNA primer at the 3' terminus of the lagging strand, DNA polymerase is not able to replicate this DNA when the primer is removed.
Now, see what will happen at the end of the DNA. See, there is a primer which is being added. This primer will provide the will provide the template for the DNA polymerase to replicate this fragment of the DNA. But what about the fragment of the single stranded DNA that will be produced after removal of the primer? What about this particular fragment? This will remain a single stranded DNA and will not be replicated. So will be subjected to degradation by exonucleases. Please have a look here.
This leaves a short region of unreplicated single-stranded DNA at the end of the chromosome.
See, this has been replicated, this has been repaired, but what about this part of the chromosome? This part of the chromosome cannot be replicated because there is the RNA primer which is present. Now you have to attach a primer somewhere here, but there is no template. You cannot add a primer. Then what will happen? See, this is the single stranded DNA that has remained. This is the unreplicated single stranded DNA. And it is this DNA that will be the subject of action of exonuclease, which will begin to cleave the DNA molecule, subsequently degrading the DNA molecule into shorter fragments. Look here.
This incomplete replication is called the end replication problem. If eukaryotes did not have a way to solve this problem, the linear chromosomes would become shorter after each round of replication, leading to loss of genes at the ends of chromosomes.
Now see, this is the major problem that is referred to as the end replication problem. Now what is going to happen in the case of the end replication problem is that that the incomplete replication of the 3' terminus of a template DNA molecule is being done. When the incomplete replication or the 3' terminus of a template DNA is being done, then it is caused by the 3' to 5' activity of DNA polymerase. The activity of the DNA polymerase is being lost. Okay. Then there is a loss in this entire structure. See, firstly, the 3' end is being lost here. You can find it out here that this fragment, this one was the original DNA. This is original DNA. Once this is the original DNA that is present, this original DNA loses its structure. Okay, this is its structure that would be lost. It would be degraded. This has been degraded. Then on further degradation, what is going to happen? This fragment is going to be degraded. When this fragment is further being shortened, then the other fragment will be degraded and so on. So how much amount of DNA has been lost? You can look here from here till here. Lost DNA. This is the amount of DNA lost. DNA lost. Now this is the DNA which has been lost due to exonuclease activity. Due to the exonuclease activity. The exonuclease activity. This is the exonuclease activity that has lost, that has caused the loss of the segment of genes which were being associated with the telomere. Now these has to be protected, else the integrity of the genome will be lost. Now how they will be protected, that we will be looking forward, in what manner the protection has to be done. So kindly look forward.
The ends of eukaryotic chromosomes are called telomeres, and they are generally composed of repeats of a TG-rich DNA sequence. For example, the sequence TTAGGG is repeated many times in human telomeres. The 3' end of each chromosome extends beyond the 5' end as single-stranded DNA. as would be expected due to the end replication problem.
Now see why this 3 prime end is being extended in such a long manner. This 3 prime end actually represent the area where the primer was being held. Okay, it was actually a primer. It was actually a region where the primer was present. It in this particular region, this one was the primer which was providing the priming activity for the DNA biosynthesis. It was providing the 3 prime primer, okay, through which the DNA has been biosynthesized. Now, this primer has been degraded with the help of RNase H. You know that RNase H? This is RNase H. Okay, RNase H was the protein that was binding to this particular site, was degrading it, was fragmented it, and then this RNA sequence, that one, this which was referred to as the primer RNA, was degraded. Primer RNA was degraded. RNA was degraded because primer RNA was degraded with the help of RNase H. So there is a gap that is going to be produced. Okay, moving forward, that how this gap will be repaired.
Most eukaryotes use a specialized DNA polymerase called telomerase to solve the end replication problem. Telomerase is comprised of both protein and RNA. The RNA includes 1.5 copies of the complement of the telomeric sequence. For example, human telomerase RNA contains the sequence UAA-CCC UAA.
Okay, there is a telomeric sequence that is present. UAA, CCC, UAA. This is a telomeric sequence that is present over the RNA molecule. Now, to this telomeric sequence will provide the template for the biosynthesis of the complementary DNA molecule. Okay. Now, see, this is an RNA dependent DNA biosynthesis, but there is no template. So, telomerase catalyzes a template independent DNA biosynthesis. There is no template here now. So, it will synthesize a new template. It catalyzes template independent DNA biosynthesis using its own template. Okay.
Like all other DNA polymerases, telomerase extends the 3' end of its DNA substrate. However, unlike most DNA polymerases, telomerase does not need a DNA template to direct the addition of new nucleotides. Instead, the RNA component of telomerase serves as the template for adding the telomeric sequence to the 3' terminus of the chromosome.
See, this is what is going to happen. Telomerase will bind to the ends of the chromosome, but will use its own RNA sequence to biosynthesize the DNA molecule in adjacent to its RNA sequence. So the RNA sequence of the telomerase themselves will be used in order to biosynthesize the DNA molecule.
After telomerase synthesizes DNA to the end of the telomeric template region, the RNA template disengages from the DNA product and reanneals to the end of the copied DNA. This process can be repeated many times, extending the 3' end of the chromosome.
See this. This is the process that is being repeated many number of times, extending the 3' end of the chromosome. Now, this is where the 3' end has been extended. Now, it is this DNA molecule that will serve as the site for the biosynthesis of the complementary primer, and this part of the DNA will be replicated.
The lagging strand DNA replication machinery can then extend the 5' end of the chromosome. Although DNA polymerase is still unable to extend the 5' end all the way to the end of the chromosome, the telomeric repeats protect the genes at the end of the chromosome in two ways: by serving as a buffer of non-coding DNA and by binding proteins that prevent the degradation of the ends of the chromosomes.
Okay. So it would be the one that would be binding towards the ends of the chromosome, and thus will be the one that would be protecting the ends of the chromosomes from the degradation. So it would be the one that would be protecting the ends of the chromosome from the degradation process that is going to take place. So the degradation will be prevented, and here the ends are being replicated effectively. Now this end is going to fold, and when it is going to fold, Along with the telomerase binding protein, it will form a loop shaped structure, and that would be referred to as the T loop and the L loop. Now, how loops are being produced, that we are also going to see.
Telomerase
See, and now please have a look here. I would be just showing it to you. Telomerase loops. Okay, I'm just going to show it to you, the process of loop formation. Please have a visualization here. Let me move to that particular site where it is given. It is from a research paper that I would be showing it to you. Just a second. Okay, moving forward. Look here. This is the G strand, this is the C strand. Please pay attention over this particular uh, diagram which is being shown to you. In this particular diagram, you can see this. There is the strand of the DNA that has been replicated and extended. See the blue strand of the DNA. Okay, after the extension has taken place, this blue strand is further penetrating the C strand, forming a complementary copy over itself, and now the telomerase proteins are going to act. So here now the telomerase protein, now the telomerase binding protein will act. How? It will bind to this particular location. This would be the area where telomerase binding protein will act. Okay, TBP. Okay, telomerase, this is the area where this protein is going to bind, and this protein will be commonly referred to as the telomerase binding protein. Just a second, sorry, this would be referred to as the telomerase binding protein. Here also the telomerase binding protein is going to act, and both these proteins will effectively ensure that the DNA forms the two loops, the T loop and the D loop, and this would be the, these two are referred to as telomerase binding protein. Telomerase binding proteins. Okay, binding proteins that are being produced. Any doubt? If you have, if you have any doubt, you can just ask me. Okay, this is how the telomerase are going to fold, and in this manner the formation of the T loop and the D loop takes place, not only on one end of the chromosome but on both the ends of the chromosome. Okay, if anyone has any doubt, you can always ask me up or comment in the comment box. Now moving back to the Pathfinder, where we were talking about the different types of modifications. Okay, so now moving back to Pathfinder, this is how the ends of the chromosomes are being replicated. Now another type of replication that is of extreme importance is referred to as the rolling circle replication. Now rolling circle method of replication. Rolling circle method of replication is a method of replication that takes place in the case of phages. What happens in the case of phages? Because phages have to replicate at a much faster rate, as the phages as sorry as phages have to replicate at much faster rate. Just a second, just a second. As phages have to replicate, have to replicate at a much faster rate, at a much faster rate. So they have developed, so these have developed a unique, so these have developed a unique method, a unique method in order to, method in order to replicate their DNA, to replicate its DNA sequences, its DNA sequences via rolling circle mechanism of replication. The mechanism of replication that the virus is using is referred to as the rolling circle mechanism of replication. Circle mechanism of replication of DNA replication. Okay. Now, what happens in the case of rolling circle mechanism of DNA replication? The DNA is going to be replicated in a circular manner. There is a cleavage or a cut is being produced at a particular site. Once the cleavage is being produced at a particular site, the template in circular double-stranded DNA molecule, there is a cut or a cleavage which is going to be produced. When the cut or cleavage is going to be produced, then what is going to happen? It will create a 3 prime end and a 5 prime end. This 3 prime end will be extended further. Okay, it is this 3 prime end that is going to be extended. See this, the primer is being used and here it is being extended. Here it has been extended, 3 prime end and extended. When it has been extended, it will replicate over the other complementary DNA molecule and the pre-existing DNA molecule will be replaced. It would be chopped from here, it would be cleaved from here, and will be the one that would be again forming a particular circle. Okay, it would be the one that will be forming a particular circle in this manner. Now further on further movement, it will form a particular circle. The circle will be completed and then degraded, formed another virus particle. It will form a new virus particle. This is how virus particles are being generated. How virus particles are being generated? Because for every time the biosynthesis takes place, a new circle is being produced. Once a new circle is going to be produced, then ligation will take place. And once the ligation takes place, it will form the circular DNA molecule. This is the circular DNA molecule that is being biosynthesized. This is how the biosynthesis of the circular DNA molecule takes place. Okay. And now there would be a primer. This primer will be extended in the following direction. When the primer is going to be extended, the primer will lead to the formation of a double stranded DNA molecule. This would be the primer. Primer will be extended, and this will lead to the formation of a dsDNA. This is the dsDNA molecule that is going to be produced. Any doubt you have, you can ask me. Okay, this is how rolling cycle mechanism of replication takes place. See, another method is the rolling circle mechanism of replication that generates linear copies of a genome rather than its circular copies. So there are linear copies that are going to be generated in this type of replication machinery. What is being done? Consider a circular molecule of a double stranded DNA molecule. For example, there is a circular molecule of the double stranded DNA. There is a nick in one of the two phosphodiester backbones. There is a cut. There is a nick in one of the two phosphodiester backbone. When the nick is being made, it provides a primer for the action to take place. The nick opens one strand and the 3 dash strand of the OH that is being generated will act as a primer. The primer will be extended. As the primer is being extended, the 5 dash end is being displayed in a manner analogous to the strand displacement reaction. The extension of the 3 dash OH is also analogous to the leading strand synthesis. The displacement strand can in turn serve as a template for the replication as long as suitable primer is available. As long as the primer is being available, there is the biosynthesis taking place. Okay, synthesis on this strand is analogous to the synthesis on lagging strand. Such type of replication is found in M13 phages, in filamentous phages such as F2 phages, F12 phages. Okay, the biosynthesis leads to the formation or production of the conchimers copies of the circular DNA molecule. There are continuous copies of the circular DNA molecule that is referred to as contamer copies of the circular DNA molecules that are going to be produced. Okay, which are the copies? Contamer copies of the DNA molecule which is being produced. Okay, as a result, multiple copies of the genomes are being produced. A rolling circle mode of replication is seen both in replication of bacteriophage lambda as well as in the case of M13 phage. So there is bacteriophage lambda where it is being found. There is M13 phage where it is being found. So these are the two different organelles which depict the rolling circle mechanism of replication. Now, the last mechanism of replication that occurs in the case of mitochondria, that is referred to as the displacement model, also referred to as the D loop model of DNA replication. What happens in the case of D loop model of DNA replication? In this model, what is being done? A single D loop is found as an opening of 500 to 600 bases in the mitochondria of the mammal. Now, what happens? There is a single loop that is going to be produced. A single D loop is found as an opening of 500 to 600 bases in the mammalian mitochondria. Okay, there is an opening which is being found, and that opening refers to the D loop. Now, once the opening has been made, a primer is being provided. Once the primer is being provided, the primer is being extended. See this? DNA synthesis initiate at origin on the H strands of the DNA. On the H strand of the DNA, there is a primer that is going to be added. Okay, this is the primer, DNA primer or the RNA primer. RNA primer. This primer is being made to extend with the help of DNA polymerase. Okay. Synthesis of new L strand creates D loop by displacing the parental strand. There is the parental strand. The parental strand has been displaced. There is a loop which is going to be produced called the D loop. You can look here. This represents D. That is why it is referred to as a D loop. There would be a loop like structure produced, and that would be referred to as the D loop. When displaced strand passes origin on L strand, synthesis of a new H strand takes place. When the displaced strand is being passed, there is the biosynthesis of the new H strand that is going to take place. Okay. So when displaced strand passes origin on the L strand, synthesis on the new H strand is going to go through. There is a new biosynthesis that is going to take place, and here the DNA is going to be duplicated. The two genomes of the mitochondria will later on get separated from each other. These two will be separated. How they will be separated? These two will be separated in the following manner. There would be a double stranded DNA molecule here. Okay. There would be another double stranded DNA molecule. See this? This is a double stranded DNA molecule. Another molecule will be produced in the form of a single stranded DNA molecule, and that single stranded DNA molecule will undergo further replication. It will form another single stranded DNA molecule. Okay, look here. And this single stranded DNA molecule will provide the template for the biosynthesis of a new DNA molecule. I'm making it in dotted form in order to make you understand that this is the site where the new DNA is being biosynthesized. So this is parental DNA, parental DNA, and this is new DNA biosynthesis, new DNA molecule. This is the new DNA molecule that has been biosynthesized. Is this clear to you? Is this clear to you or not? This is how the entire process is going to take place. So this will synthesize the new strand of the mitochondria and new DNA of the mitochondria. Thus, the mitochondria is going to be divided into two. The DNA in the case of the mitochondria is commonly referred to as cccDNA. This is called as cccDNA, and there would be another copy of this cccDNA because this is referred to as circular covalently closed DNA molecule. It is commonly referred to as circular covalently closed DNA molecule, closed dsDNA molecule. Okay, this is where the circular covalently closed DNA molecule is going to be produced. Okay, So this is all over. We would be starting with recombination in tomorrow's morning class at 10:30. I will sharply start with the recombination, and then in evening we'll be finishing off the recombination process. Is this clear to you? If you have any doubt, you just ask me. That's all for today. Thank you.